topics for AS, which is actually really good. So for year 12. So this is sort of the last little bit. We won't get it obviously all done today, but couple of weeks into August, by the time we get to September, so those first 2 weeks in August, we should get most of this done. The main thing that probably will take a little bit longer, and I won't start it today, is the statistics side. So there's lots of statistics that you can get asked to use and understand in the exam when it comes to doing the exam, that basically backs up a lot of ecological biodiversity and also genetics. So basically just statistics to back up, are these actual results actually worthwhile or not? Are they something that we can actually prove are actually worthwhile, or is it just by chance? So I might mention them roughly today, but then what we'll do is we'll probably spend a couple of sessions in September just really working through the statistics and basically how we do them, what we need to do, and what we really need to understand with them. They're not really too hard. People sort of get themselves a bit worried about them, but actually it's, I think, just mainly because it's completely new concept, but actually really the statistics are really just more an extra added part that we can sort of show. It's very mathematical. It's very much, just knowing a couple of rules, and once you understand those rules, then you can sort of prove in an answer. You'll often get given some experimental data, and you need to prove if it's by chance or there's actually a significant difference. So you can use statistics you learn for that, and that's basically it. But why I'm mentioning that is it sort of comes into this topic. So the first sort of bit of topic is we're looking at classification. Again, this used to be a completely brand new topic at A level, but it did come into the GCSE. So again, lots of this will be slightly familiar, maybe with a little bit more extra information. So classification. Look at what classification is, history of classification, and taxonomic hierarchy. So classification is our arrangement of organisms into groups of various sizes and based on shared features. So basically, somebody came along and sort of went, well, we've just got all these animals, got all these plants, we've got all these living things, and we sort of wanted a way to classify them so we could say, actually, this is a plant, this is an animal, this is non-living. Straight away we wanted to group things like that. We group things naturally as humans into living, non-living, then we get living things and we group them again. It's a very natural thing to want to do, just because it's actually easier for us to understand things, so we can sort of generalize different plants, different animals, because they belong in certain groups. So that's what the idea of classification is. So here's an example of a genealogical tree of humanity. Taxonomy is a form of classification. So classification is just basically grouping things. Taxonomy is a type of classification, and taxonomy focuses on physical similarities between different species, and this is for ease of naming and identification. What we'll probably actually cover a little bit is actually now we're bringing in genetic taxonomy, and lots of the judicial taxonomy is sort of changing, and we're actually finding things that might look very similar aren't actually genetically very related. We also have phylogeny, and phylogeny is the classification of organisms by their evolutionary relationships, and so that's every group shares a common ancestor. So taxonomy and phylogeny can go together, and again, phylogeny is changing due to our increased understanding of genetics. So what you might get to do is you often get asked things like, well, how might you classify these animals? And there's lots of things people could do. They look at these animals and they could go, oh, well, we could stick platypus, dolphin, turtle together because they're all in water. Oh, and a fish, I missed the fish. Well, crab can also go in water, so we can put that together. And we can basically, if we can justify why, we can sort of classify animals in any sort of groups, and we can look at the distinguishing features. So again, which of these species could be classed as aquatic organisms? And this is sort of how people came up with the classification idea originally. Well, we go, well, a fish could, platypus could, dolphin could, turtle could, and I guess we can go with a crab. I don't know if it's going to let me say crab. Yeah, it will. So you could group them all as aquatic organisms, those ones, and say, okay, well, the difference between these organisms is aquatic and non-aquatic. Then you could say, okay, which ones are fur covered? So then you'd go, okay, well, I guess we'd say platypuses, a kangaroo, a bat, probably leave it there. Then we could say which one's got exoskeletons. So we've obviously got a crab, a hood-nailed beetle. Oh, what's happened? Yes, I was trying to click that. Okay, which produce young by laying fertilized eggs surrounded by a protective shell. So you can sort of get the idea of where this is going. So we can group them all by different things, and lots of them overlap. So protective shell, go platypus. Oh, actually, no, not platypus. No, turtle. I've missed one out. There we go. And which are capable of winged flight. Winged flight. There we go. Got 2. Oh no, bat 3. So again, we've the idea of that. Oh, echolocation. We could go dolphin, and we could go bat. And the idea of this is actually we can sort of group any animal sort of anywhere that we want to, because they all share lots of common characteristics. So we're going to look at Linnaean phylogeny, taxonomy, first. So there's many different varied ways of classifying organisms. Those ones that we just talked about, they'd all be acceptable ways of classifying an organism. Yes, all those organisms, those ones were ones that lived in water. These ones all were ones that could fly. But what it is, it's important to be able to choose key features and we ignore others. So for instance, many insects and birds are capable of wing flight, but the two groups are very different, so we don't merge them. So the first scientist to introduce a formal system of taxonomy was Carl Linnaeus, and we call it the Linnaean taxonomy. And it's actually still the basis of the systems we use today. It might have changed and adapted over time, but it is that original basis that we use. So the evolution of classification, we're just going to have a quick look at that. So we're going to look at where it sort of started and where we sort of ended up at now. So back in 1735, we basically had a 2-kingdom system. So Carl Linnaeus was the first to propose a formal system of taxonomy, and his systems basically comprised of 2 kingdoms. He had Plantae and Animalia, which obviously were plants and animals. So then in 1866, we got the 3-kingdom system, and that was due to Ernst Haeckel, and he extended the kingdom to include a third, and that was Protista. So this is a group of unicellular or very basic multicellular organisms. So then we had the 3-kingdom system. Then we got 2-empire systems. So Eduardo Chatton, he recognized the basic classification into the empires of eukaryotes and prokaryotes. And he distinguished between organisms whose cells have a nuclear membrane, so our eukaryotes, and those without, which were our prokaryotes. So then he split them off again. But you can see they're all coming from that same basis, and we're just adding as we gained and learned more information. So then we got a 4-kingdom system. So the guy before, he decided to put them into 2 empires. The next guy had the same 4 groups, but he did it as 4 kingdoms instead. So Herbert Copeland reverts to Haeckel's kingdom model, but adds a 4th Monera to the system. Monera comprises all the prokaryotes of Chatton's system, distinguishing them from eukaryotic protists. Then in 1966, we had a 5-kingdom system. That was due to Robert Whittaker, and he identified a 5th kingdom of fungi, and they show a different cellular structure from plants, so they came into their own group separate from plants. 1977, 6th kingdom system. Carl Rose is the first biologist to employ phylogenetic taxonomy of ribosomal RNA, and he actually came along and he separated Monera into 2 separate kingdoms, and that was Eubacteria and Archaea bacteria. So that meant we had 6 kingdoms in total. And then in 1990, which is the one that we sort of use now, we have our 3-domain system. So Vos refines his system and he combines it with Chatton's idea to introduce the 3 domains. So we have the 3 domains of Eubacteria, Archaea bacteria, and Eukarya. And then inside Eukarya, we have Monera, fungi, and sorry, we have protists, fungi, animalia, and plantae. So that's our 3-domain system that we use today. So how are organisms classified in modern taxonomy? So we're going to have a quick look at that. So we have lots of different levels, and we go from subspecies up to superphylum. So the following 8 levels are what we widely accept as our modern standard. So we have domain. And this is our highest taxonomic group. There are 3 domains. We have eukarya, eubacteria, and archaebacteria. All the species pictured belong to the eukarya. Then we have kingdom. This was originally the highest taxonomic rank, but now we usually consider it to be 4 kingdoms within the domain eukarya. So then we have kingdoms inside the domain eukarya, and they are Animalia, Plantae, Fungi, and Protista. Then we have phylums. So the pictured animals all belong to the same phylum, Chordata, which are the chordates. So this includes all vertebrates as well as animals with a primitive spinal cord known as notochord. So obviously there's lots of different phylums, but we're just looking at one specifically. So we go domain, kingdom, phylum, and then from phylum we go to class. So we could have the class Mammalia, and this comprises animals that produce milk for their young from mammary glands, and other examples of other classes are things such as insects and crustaceans, and they would come under the phylum Arthropoda. So under a different phylum, and then you'd have the class of insects and crustaceans. Then we have orders. Sorry, one second. Sorry, my hay fever is just really bad today. So we've got primates. So these are an example of an order. This group includes new and old world monkeys and the great apes, as well as lemurs and tarsiers. The rodents are another mammalian order for an example of where we could sort of go off to. Then we have families. A family is a relatively small taxonomic group. So humans, chimpanzees, gorillas, and orangutans together form the family Hominidae, and the hominids are great apes. Then we get genus. A genus is the smallest grouping of species. Homo sapiens is the only living species belonging to the genus Homo. We have obviously had others, such as Homo ergaster and Homo neanderthalensis, and Homo erectus. So obviously we've now just got Homo sapiens and then species. So the Homo part would be the genus, and the species part is the second part. So that's a binomial name, so it's a 2-part name. A species usually consists of a group of individuals, and this is really important definition because this question almost always, I can't say it always gets asked, but it gets asked a huge amount of time. What is the definition of a species? And often people write all weird and wonderful things, but basically a definition of a species is a species consists of a group of individuals who can successfully reproduce to create fertile offspring. And it's that term fertile that's really important. Individuals not of the same species can reproduce to create offspring, but they're not fertile. They're usually hybrids that are infertile. So that would symbolize that their parents have come from different species, similar enough to create offspring, but actually not from the same species because their offspring are infertile. So the definition for a species is groups of an individual who can reproduce to create fertile offspring. Species names must always include the genus they belong to. So for instance, Homo sapiens. And also notice that they're in italics and the genus is in a capital letter. So the Homo for the genus, the H, whatever it is for the genus, the first letter of the genus is always in capital letter. Species is never a capital letter. So these are little rules in writing them. If you're handwriting them, we underline them. If you're typing them, we use italics. It's just sort of some rules that sort of come into when we use the actual scientific Latin names for species. So quick overview of that again. Homo represents the genus, sapiens represents the species, but we always write the species name with the genus name. Genus always has a capital letter at the beginning, species never does. If we're typing it, we use italics. If we're writing it, we underline the word instead. So Plantae. So the kingdom Plantae. So the main characteristic feature of individuals in the plant kingdom is their ability to manufacture their own sugars from simple molecules by photosynthesis. So they're known as autotrophs. The other main defining feature is their cellulose cell wall, and that is found in every cell. And There are an estimated 350,000 species of plants, which include trees, herbs, bushes, grasses, vines, ferns, and mosses. We've got the kingdom Animalia. So members of the animal kingdom are multicellular heterotrophs, meaning they depend on other organisms for food. All animals, except sponges, have a nervous system, a muscular system, and it enables them to react quickly to environmental stimuli. This means most animals are motile. They can move spontaneously and voluntarily. Sponges actually used to exist under the Plantae kingdom, but they got changed as more research went into them, but people actually realised that they're not plants. They're not actually an autotroph. They are a heterotroph, but they don't have the same nervous system as every other sort of animal would. The animal kingdom contains most multicellular organisms and is the most diverse of all kingdoms. We've got kingdom of fungi. Fungi are eukaryotic heterotrophic organisms that have a chitin cell wall. Again, sponges didn't come under this because they don't have a chitin cell wall. Some fungi grow as a single cell, but most are multicellular, and they reproduce via spores. And again, sponges don't reproduce this way, so often produced by specialized fruiting bodies, and this is called the sexual phase of a fungal life cycle and is the most visible. Mushrooms, for example, of the fruiting body, so the whole mushroom that we see is the fruiting body. We actually have a lot more of the fungi growing under the ground that we can't see. So the vegetative part of a fungus consists of a mass of thread-like structures called mycelium, and this often extends and can reach a massive size underground. Then we have kingdom Protista. Protists are a group of eukaryotic organisms that do not fit into other kingdoms. As such, they don't actually have much in common with each other, apart from having a relatively simple organisation. They're unicellular or multicellular, but with unspecialized cells. Some groups resemble other kingdoms. So the protophyta resembles plants and protozoa resembles animals. And then we finally have kingdom Monera. And Monera are bacteria and other unicellular organisms whose genetic material is loose in the cell. They form one kingdom in the 5-kingdom system and were previously called prokarya. Monera have been subdivided into the domains Eubacteria and Archaeobacteria based upon molecular phylogenetics. This is actually quite recent. So we're talking like past 10, 15 years here, rather than hundreds of years. And then into 6 kingdoms, as well as into kingdoms in the 6-kingdom system. So Archaeobacteria initially thought to live only in extreme environments, so they were called like extremophiles, but we've actually now found them in all major habitats. I think that is it. So let's look at the next bit. So phylogeny. Okay, so what is phylogeny? When we group species according to their shared characteristics, the resulting taxonomy will often reflect their evolutionary relationships. Sometimes, however, there's a superficial resemblance arises in species from different branches of the evolutionary tree. So this can happen if 2 species move into a similar ecological niche, and we'll talk about ecological niches in a bit, or if one species starts to mimic another. So some species may also appear dissimilar when actually they're really closely related. So phylogeny seeks to classify organisms according to their true families and avoids this sort of confusion. So we're going away from just physical characteristics, we're actually looking at the genetics, actually their evolutionary past. So the evolutionary tree. So how closely are these 6 primates related to humans? Well, we'd have to say chimpanzee, probably orangutan. I'm just going to put them in any order. We know they're going to sort of be there. Old world monkeys, new world monkeys, stick a lemur at the bottom. Let's submit. Right. There we go. That's what I wanted to get to for the answer, so we could see this. So basically, here we have a phylogenetic tree, an evolutionary tree. And so we have humans here. And basically, this is the common ancestor of everybody at the bottom. 50 million years ago, there was a common ancestor and it diverged into lemurs. And then we get to this next part and it diverged again and we had New World monkeys. So if you look, there's 15 million years, that is right, yes, that is right, between the evolution of the lemurs and New World monkeys. 5 million years later, we get the Old World monkeys diverging off. Orangutans, 13 million years later, then gorillas, then chimpanzees, and then humans. It's about showing the history between them, and these parts here show the where they join is that they have an evolutionary ancestor that they would both share. So phylogeny in practice. So the key to a phylogenetic classification is we look for the sorts of common features that must be due to common ancestors and not due to evolutionary pressure. So the features that share a purpose, such as streamlined body for swimming, aren't useful because they're to respond to their environment where they live, and evolutionary pressure, an ecological niche. Whereas useless features are actually likely to be an accident that has arisen only once. So for this reason, a really good indicator of relatedness is the similarity of sections of non-coding DNA. So much of phylogeny is conducted using molecular systematics. So what is molecular systematics? So molecular systematics uses molecules such as DNA, RNA, and proteins to interpret the evolutionary relationships between molecules. So stage one, we get an organism and we will take a sample of its DNA. So for example, here we've got Clostridium bacteria. These are then amplified using PCR, so polymerase chain reaction. I think we did that. I can't remember actually if we've done that yet or not. We might have done that when we were looking back at DNA. It's the idea that we get a small amount of DNA and then basically putting the right factors in with it in a lab, we can amplify it up through this polymerase chain reaction, putting the enzyme polymerase in and free nucleotides to make larger amounts of a DNA. Basically because the sample isn't big enough really for us to do much with. We want to make it bigger. It means that we aren't in danger of wasting that one small sample because we made a lot from it. It's used all the time for things like genetic testing, for things like paternity, crime scenes, that sort of thing. So and it multiplies the number of copies of DNA in each sample. So that's our first step. So then the second step, we dye these samples and we place them in holes in a sheet of gel. And we use something, we basically go through a process called gel electrophoresis, which we will look at some point. And basically it's a gel that's got holes in, and we run an electrical voltage across this gel, and the molecules are separated out by size. So the DNA strands of molecules are separated out by size, so heavier ones don't travel as far, lighter ones travel further, and we can see them because we've dyed them. And we can actually use a different dye to stain each nucleotide, so A, T, C, and G can have a different coloured dye. Stage 3, we make a DNA barcode, and a DNA barcode is created for each organism. So this is a relatively short sequence of DNA that can be used to identify the organism, so it's unique to that organism. So DNA barcodes of all the organisms being studied can then be compared to each other. So we can look at the similarities. So for this example, they're all in the genus Clostridium, and then we can look at them and say, okay, well, difficile and botulinum have similar here, and they've all got the same at the end, and we could sort of look at the similarities and differences that way in the barcode. So then stage 4 is our final process, is to order the sequences from most to least similar, and we use this to construct an evolutionary tree. So the most similar in this example was C. difficile and C. botulinum, which is here. So these two were our most similar, and we can see that quite easily. So because they were the most similar, they were most closely related, so they'll have evolved more closely. So it means that they're more ancient evolutionary ancestor would have been the Titane. So let's close that. So what is a species? Well, you've already had my definition of a species, but species we often think it's really easy to find a species. So lion and tiger, very different species. But then we can get into things where people say, oh, well, this is a Bengal tiger, this is a Siberian tiger. Are these different species? We can use the example of dogs. We use the term breed. There's a reason we use the term breed for dogs is because actually all dogs are the same species. We've just selectively bred them to look extremely different from each other. But actually, we could breed any dog with any dog and they would all produce fertile offspring. So Bengal tiger and Siberian tiger. So if two populations are geographically isolated, it can be really hard to tell if they're capable of interbreeding to produce fertile offspring. So this is one aspect of the species problem. Because they don't live near each other, we don't actually know if they could interbreed and create fertile offspring. So we don't know if they're species that way. Also, the capacity to interbreed cannot be tested in animals that were extinct. So, for example, dinosaurs. So to overcome this problem, we needed other definitions of a species. So we use the term biological species concept, and this is our most common definition of a species. It defines a species as a set of individuals who can reproduce to produce fertile offspring. As well as the problem of geographical separation, another disadvantage of this definition is that it only applies to organisms that reproduce sexually. So obviously any asexual organisms this creates an issue for. So the phylogenetic species concept defines a species by its evolutionary lineage, where two lines diverge sufficiently they're called a separate species. A problem with this definition is deciding what constitutes sufficient divergence. So binomial naming, so this is back to what we were saying before about the naming. So when a new species is identified, it's always given a binomial name. So that's those names of two parts. So for example, Panthera tigris is for the tiger. So Panthera is the genus, so we'll have loads of other species that are under the genus Panthera, because they're basically our big cats. And then Tigris is for tiger. So if a subspecies is identified, an extra name is added to the binomial species. So for instance, the Siberian tiger is known as Panthera tigris altaica, and that's because it is classed as a species, but it's a subspecies. It's, I guess, if we were sort of going with dogs, we could maybe call different breeds a subspecies. Okay, so a dichotomous key is a key that you can follow to basically work out a species. So you basically get given information, you have to say yes or no, and it will take you to the species if you didn't know what the animal was. So we've got an image here. So we'd follow the key to identify the cat species. So does it have solid colour fur or patterned fur? Well, it has patterned fur. Does it have pointed ears with prominent ear tufts, or does it have rounded ears with ear tufts? Well, it has pointed ears. Does it have a uniform tawny coat or black and white markings around the muzzle? I'll go black and white markings. Does it have a striped coat or other coat, or has an other coat? And so this is a cheetah. It is the only species in its genus and lives in parts of Africa and Iran, and is an endangered species. So that was nice, short and sweet. Biodiversity now. So we've sort of covered what a species is, classification, looking at biodiversity. This, again, will have been slightly touched at GCSE, but now we are going into a lot more depth here. So let's have a look. So what is biodiversity? So biodiversity, we basically talk about how diverse an area is. So we can look at areas that have low levels of human expansion. So if you work down through the tab, see what happens to biodiversity as a result of human expansion. So here we've got an image of a really biodiverse environment. And as there's more human expansion, for example, we take away some trees, the diversity should start to decrease. We take away more trees, the habitat behind gets taken away, we've got a reduction in species again. And as you can see, reduction in species continues until we have that. And that's our high levels of human expansion. So what is biodiversity? It is a measure of how varied an ecosystem is. And it can be measured in three ways. We can measure in terms of genes, in terms of species, or in terms of habitats. So genetic diversity is a measure of how many variations there are in the genetic code between individuals of a particular species or between different species. So basically, if we're looking at one population, how genetically diverse are they? Are they all actually really closely related? Is there not much variation in the DNA, or is it actually quite a diverse population? Or we can look between species at the genetics as well. We can look at species diversity, and that's just a measure of how many different species there are present in a specific area and how many individuals of these species there are. And that's really important. We can't just look at how many species there are, because you might have an area that's got a really high species diversity, species abundance, but actually the diversity could be low because they might have 100 different species, but only one of every single species, where you might have another area that's only got 70 different species, but they're actually quite highly populated. And then habitat diversity. So how many different habitats are there in a present area? So species, again, what is a species? Is a group of organisms that share a common morphological, physiological, and behavioral characteristics. They can interbreed to produce fertile offspring. A hybrid is when different species reproduce and they produce a hybrid. The parent organisms are still considered separate species because their offspring are infertile. So, for example, we have the liger. This is the infertile offspring of a male lion and a female tiger. Another example is the zonkey, and that's from the infertile offspring of a male zebra and a female donkey. So species diversity can be strongly affected by changes in species habitat. At the simplest level, we say a habitat is the area in which a species lives. However, it is a lot better to describe the habitat of a population of species. A better explanation of the habitat is the range of physiological, biological, and environmental factors within which a species can survive. So you're basically breaking it down to the abiotic and biotic factors of an area in which those species can survive. So habitats of different populations often overlap. So any one habitat usually contains several species. So let's use the example of a habitat of an oak tree. Bears can also be present in the habitat of an oak tree, but also in a different area. Squirrels, you might find them in the habitat of an oak tree, also in the habitat of bears, but also in their own little habitat. So a community is all the populations of all the species within a particular habitat. So we have endemism. So endemism is a reference to a group of organisms that are unique to a particular habitat, and they are geographically discrete, so such as an island or a certain type of vegetation. So we don't find them in lots of places, so they're endemic to one particular place, one particular habitat. So for example, kiwis, they're a family of birds endemic to New Zealand. And while the orange-breasted sunbird is endemic to fynbos vegetation, a type of shrubland in the Western Cape of South Africa. The opposite to endemism is cosmopolitan distribution. I think we've done that. So, why is biodiversity important then? So, biodiversity hotspots. These are areas of the world that have a really high levels of biodiversity, but they are threatened with destruction. So ones that straight away come to people's minds are things like the coral reef, the Amazon rainforest. We have 34 biodiversity hotspots across the globe, and they all contain at least 1,500 endemic species of vascular plants, and which have lost 70% of their original habitat. So remember, endemic means they're only found in that specific habitat. So again, things like mangrove forests are often biodiversity hotspots as well. So these hotspots account for only 2.3% of the total land area of Earth, but they contain at least 50% of all plants, 42% of all terrestrial vertebrates, and 29% of all freshwater fish species. So we're going to have a quick look at some of these biodiversity hotspots. So California's floristic province is one of them. They've got endemic threatened birds, they've got 4. Threatened mammals, they've got 5. Threatened amphibians, they've got 8. Extinct species since 1500, they've got 2. The Caribbean islands, again, they've got endemic plants, 6,550. Birds, 48. Mammals, 18. But we've had 38 extinct species since 1500. Madrian pine oak woodlands. We've got Mesoamerica, Tumbes choca magdalena, tropical Andes, Cerrado, Atlantic forest, the Chilean winter rainfall, the Valdian forests, done tropical Andes, Mediterranean basin, the Guinean forests of West Africa, the Caucasus, Irano and Anatolian, mountains of Central Asia, Himalayas, mountains of Southwest China, Indo-Burma, Japan. You'll notice lots of these are islands as well, and you'll sort of start to work out why lots of them are islands, because of that endemism that can occur. Probably would have clicked on this, but yeah, we did. Guinean forest of West Africa, succulent Karoo, Cape Floristic region, Maputo-land, Pondo-land, Albay, Madagascar and the Indian Ocean Islands, coastal forests of East Africa, Eastern Afromontane, Horn of Africa, I've done that, Western Ghats and Sri Lanka, Indo-Burma, Sunderland, Philippines, Wallacea, which is a really, if I put it on here actually, Wallacea is really tiny. Polynesia, Micronesia, East Malaysian Islands, New Caledonia, New Zealand, and Southwest Australia. So why are these important? Why do we actually care about them? So low biodiversity isn't necessarily cause for concern. So the Arctic and Antarctic contain habitats that naturally have a low biodiversity compared with tropical rainforests, just because they're very extreme. What's more important is the loss of biodiversity. So an ecosystem relies on the interdependence of organisms to maintain its stability. And as we start to lose species, this can cause serious, unpredictable, and possibly irreversible changes to that habitat. So ecosystem disruption can ultimately have a significant impact on humans. So I'm going to look at hedgerows. Hedgerows are something that we see quite a lot in the UK. And they're fantastic. They're actually home to lots and lots. They're home to several habitats, and they can support many different types of insects, birds, small mammals, plants, and flowers. We're starting to see a lot more hedgerows reappear, but there was a huge, probably about 50 odd years ago, there was a huge upsurge of destruction of hedgerows up until recently as well, and that was to increase the productivity of arable land. And basically farmers wanted larger pieces of land. It wasn't that they didn't own them, they owned the land, but the hedgerows would break up a piece of land. If you don't have the hedgerow there, you don't need to move a tractor around, and so on and so forth. So a lot of hedgerows are removed. However, that's led to a huge decline in these organisms that live there. They can actually be essential for maintaining the balance of that ecosystem. So without these, food webs can actually fall apart, diseases can spread rapidly, and crops can become more susceptible to pests. So low biodiversity in itself can be problematic in some situations, such as agriculture. So we can get selective breeding coming in. So this is when we have that monoculture with monocrops. So this is when we create crops with desirable characteristics, and they are very similar. So individual plants, therefore, have similar growth patterns, and it's great because it leads to higher yields and less wastage. However, this lack of genetic diversity can make the crop as a whole highly susceptible to pests and diseases. If a new disease comes, if a new pest comes to that area, it can wipe out that whole crop. So in agriculture, just growing one species variety is called monoculture, and we're trying to avoid promoting monoculture. Whilst it's really great economically, it can have these really negative downsides to it as well. So economic effects of agricultural monoculture can be illustrated by devastation of the southern US's cotton industry in 1920s and 1930s. This was called the Dust Bowl. So in the early 20th century, many southern states of the USA grew cotton, but they depended on just one variety. And what happened was they had the migration of a boll weevil, and it was an insect that feeds on cotton plants. So this came from Mexico to the US between the late 1890s and 1920s, and it was absolutely disastrous. What happened is it caused massive crop losses, and it contributed to the Great Depression of the 1930s in America. So, how do we measure biodiversity then? So why do we want to measure it? It's great because it helps us compare different things. It helps us compare different areas. So, for example, we compare the biodiversity of one section of woodland with that of a similar type of woodland in a different geographical area to see which requires the most protection. So, you could, for example, look at the woodland at a wood in Leicester, and you could look at a woodland in a wood in Nottingham. And as long as they've got the same sort of type of woodland, we can see which one would need the most protection due to the biodiversity of those places. It also helps us compare the same area at different times. So, for example, comparing the biodiversity of a group of fields before and after hedgerows have been removed or replanted, or the biodiversity of wood in summer and winter. So when we measure the species diversity of a particular area, an important value is called species richness. And this is the number of different types of species in a particular area. The greater number of species, the richer the area is said to be. However, this is like I said before, this doesn't take into account the number of individuals of a particular species. So a species with just one individual carries the same weight as a species with 100 individuals. So you can see how this can be skewed quite easily. So we don't just look at species richness, we also look at something called species evenness to sort of help that skewing that comes from species richness. So species evenness compares the size of a population of different species within that area. So species diversity within an area increases as both species richness and species evenness increases. So basically what you could find is you could have a plot of land, you could have a woodland that has a slightly lower species richness. So we've got woodland A and woodland B. Let's do this actually. Yeah, woodland A has 100 different species, woodland B has 75. Woodland A might have a lower species evenness rating, so it could only have like 70%, for example. B could have higher. So because of that, B would actually be even more diverse for species diversity because of having that higher species evenness. So to compare biodiversity, the areas have to be sampled to obtain the number of abundance of the species. So looking at this, we've got field 1 and field 2. They've both got exactly the same species richness. They've both got 3 different species. They've both got the same total number of species, same number of total individuals. However, field 1 has a lot more evenness going on. We've got 300 buttercups, 345 dandelions, 355 daisies. Whereas field 2, we've got daisies really basically holding the monopoly with a few buttercups and a few dandelions. So the most biodiverse field would be field 1 in this situation. So field 1 has more species evenness than field 2, so we would consider it to have the greatest biodiversity. So diversity can also be quantified by statistical methods, and one of which is Simpson's Index of Diversity. So we're going to have a little talk about that now, and then we'll revisit that again in September. So this is Simpson's diversity of index of diversity even. N symbolizes the total number of organisms of all species. So that's capital N. Lowercase n is our total number of organisms of a particular species. D is diversity index, so that's the probability that 2 randomly selected individuals belong to the same species and group. And the lowest possible value of D is 1. So the larger the value of D, the greater the diversity. We can also look at it in a different way, but we will cover this at another point. Actually, I'll have a quick run through of it here. So basically, using Simpson's diversity index of diversity, I don't know why I'm getting it the wrong way around today. So the table shows us number and abundance of different species in an area. So here's our equation, and this is our table. So N obviously, as we've said, is our total number of organisms of all species. So we work that out first. So N would be 6 plus 3 plus 12 plus 4 is 25. Therefore, N brackets N minus 1 would be 25 multiplied by 24, which would give us 600. Then we need to do the next part, which would be lowercase n brackets lowercase n minus 1. Lowercase n, remember, is our total number of organisms of a particular species. So this is a bit of a longer process. So we'd basically do that for species A, and we'd put this part here. So species A would be 6 times 5. So n multiplied by n minus 1. So basically 6 multiplied by 5, 30. 3 multiplied by 2, 6. 12 by 11, 132, and then 4 by 3 would give us 12. So in total, that would be 180. So then we'd get our two numbers, 600 from before, 180, divide them, and our Simpson's index of diversity would be 3.33. So here we would do 6 multiplied by 5, which would give us 30. 6 multiplied by 5, which would give, for all the same, which gives us 30 for all of them. Right. To get n, to get that part, we're going to have a 369, 90, 6, 12, 18. So n equals 18. 18 minus 1 be 17. I'm not even going to try to do 18 multiplied by 17 in my head because that would be absolutely crazy. It was 306. So then we're going to do 306 and we're going to divide that by 18. Should give us a diversity of a number that doesn't exist on our list. Let me check if I've done it right. 6, 12, 18. 18 times 17. So 306 divided by 90, 3.4. There we go. Here we go. Again, same thing. So that'd be 18 multiplied by 17 would give us 306. We do 3 times 2, which would give us 6. 1 times 0, which gives us 1. And then 6 times 5, which would give us 30. So then this part would just be all of those numbers here added together. So we would do 306 plus 6 plus 1 plus 30, which gives us 343. What have I done wrong? Oh no, 1 times 0 is 0. So it'll give us 342. N is just all our number of individuals added together. So we've got 18, 3, 1, and 6. So we've got 28 individuals. 28 multiplied by 27 give us 756. And then we're going to do 756 and we're going to divide that by 342. And that should give us 2.21. So we don't want to do that. Investigating biodiversity. So different ways we can basically find out how many species we've got. We could count all the organisms in each field. It's accurate. It's time consuming and impractical and probably actually not that believable for a result that you get. We could count the organisms in one small area of each field. This is quick, this is easy, but it might not provide an accurate representation of the fields as a whole, so you probably can see where this is going. We're going to count the organisms in several small areas of each field and we're going to calculate the mean. The advantage of this is it provides more accurate estimate of biodiversity of the field as a whole. Disadvantage, accurate results require samples to be chosen randomly. So random sampling and quadrats. So random sampling is used when it is impossible to count all our organisms within an area due to its size or time constraints. So there's different ways we can do this. We could throw a quadrat. A quadrat is a wooden or metal frame. It has a known area, so it could be 0.5 meters squared, it could be a meter squared. And we survey the organisms within its perimeter is one example of random sampling. So we look at what's in there and we write it down. Issue with this, it might seem random, but there is personal bias inside this. All parts of the area don't have to have an equal chance of being sampled because the throw might be limited to as far. They can throw the quadrat, for example. Someone might really want to lob it really far. Someone might just place it down next to them. A better way we can choose randomly is to divide the area on a map into a grid of equal sizes. And so, for example, on that one, they split it into 600 squares. And then use a random number generator to select the coordinates of a representative number of squares in the grid, and then we sample them. And each square can then be sampled as before, but we've got a more random sample. We can also use random walking. It doesn't sound that scientific, but it is a method. We choose a random number between 0 and 360 to act as a compass bearing, and then we choose another random number to determine how many paces we walk before reaching the sample point. We do that, reach sample point, take a sample, do the same thing again, reach sample point, take a sample. We can also use a line transect. It doesn't seem as random, but it's actually really useful because there might be a change in the habitat that you're looking at. So for example, there might be a stream. You might be looking at a large piece of land that maybe has a hill on one part, might have grassland on another, wood on another, water on another. So you might want to actually follow that changing of habitat across that land and see if that has an effect on biodiversity. So for example, the effect of a stream running through a field or wood. A line is drawn through the area to be examined, and any species touching the line at fixed intervals is recorded. So that would be an example of our distance along a transect, and we might be able to then see, well, we might go, okay, this area is where the water was. Oh yeah, we have different species present where the water is and where there's not water. So a line transect does not reflect the density of each species along the line, so it's only useful as a very basic analytical tool. We can use something called a belt transect. It's really similar to a line transect, but it provides more detailed data. So rather than simply just recording the species that touch the line, we then put quadrats where we would have looked at those lines. And we do them at regular intervals across the line to identify the number and density of species across the belt. So this will be our belt for transect is able to show the densities of the species present in the area rather than just presence and abundance. And then I think we can probably quickly, let me have a look what it is. No, I'm not going to try and start that now. Okay, so let me go back to here. Oops, what have I done? Stop sharing, that's what I want to do. Okay, so